And with that, it's my pleasure to introduce our speakers today, who will be telling us about phototoxicity in live imaging, the sources, assessment, and reduction of this critical issue in live cell imaging. Our first speaker is Philippe Laissue. He's a lecturer in bioimaging at the University of Essex, and he focuses on developing methods for quantitative microscopy, specifically working with low light conditions and in vivo fluorescence microscopy. And his work he's working both in biomedical as well as environmental applications for this kind of imaging. And our second presenter is Claire Brown, who's the Associate Professor and Director of the Advanced Bioimaging Facility at the McGill University in Canada. So we're really happy that Claire joins us at a very early time in her time zone. Claire is also involved, focuses on quantitative bioimaging. So this is what our two speakers share in common, and I think why they're connected on the topic of phototoxicity, working with live cell imaging. And Claire is also very active in Canada bioimaging and global bioimaging. And both of them are doing work around phototoxicity also in the context of the Quarabli Working Group 13. And with that, it's a pleasure that you're all here, and thank you very much to Philip and Claire for giving this presentation today. Please, you can get started with your slides.
Okay, thank you very much, Johanna. It's a great pleasure to be here, and yeah, Claire and I have worked on this for a while. Some of this came about when we set up a meeting during COVID, and really the whole idea is just to alert people a little bit to some of the issues that arise around phototoxicity and what we can do about it. And so yeah, we've done this double act a few times. So yeah. We're very happy to be able to present that here. And so we will be both presenting on and off. Maybe firstly, one of the things is that we would really like you to put in a couple of responses. We have a very short poll there. That link is also in the chat. And if you wouldn't mind briefly filling that out, because that gives us also an idea of how best to tailor these talks and what people are, how people go about, and how much of an issue phototoxicity is perceived as in facilities and in individual research. So we're going to give you a couple of minutes to fill that out, and then Claire can share the results with that. Shall I stop sharing, Claire, so you can show the results?
Yeah, that should be fine.
Okay, I'll stop the share for now. It's a very short questionnaire, but it's just a good way also to gauge where people are at and what can be done about it. So one of the things to mention maybe, and that has come up in these meetings a number of times, is that, of course, it's not just phototoxicity. You always have to be concerned with keeping your cells happy in live imaging in a whole number of different ways. Temperature, pH, CO2, and so on. But with fluorescence microscopy, using the light intensities that we have, it does seem to us to be one of the key factors that one does need to think about and assess when doing live imaging.
I think one of our other main goals is just to raise awareness too. So events like this are really great. I think a lot of people, you know, coming into the field may or may not know about phototoxicity, and I know for myself, I came from a chemistry background, and then I was working in a cell biology lab, and it was always get the light down, get the light down. and we were working on cell migration, which is very sensitive to light. So it was something that you were quite aware that it was an issue. So I think depending on the model system people are looking at, it may or may not be obvious, you know?
Yeah, absolutely. So that is definitely also one of the things that we wrote on the flag of our phototoxicity working group that Johanna had mentioned. That is part of the QuaRep LIMI quality assessment group, which is an international consortium, and we have the phototoxicity working group within there. If that is anything that you would be interested in joining, I will have a link in there later on, and you're very welcome to join QuaRep and one of its many working groups. We are working group 13 in there, and we tend to have one meeting per month, and we're slowly updating this as we go along and getting the input of many other experts in the field.
So I think we have about 16 responses, so I'll go ahead and share, but I'm happy to pass along the final results. If anyone's still working on your answers, please go ahead and we can share the final survey results with everybody afterwards. So our first question was, do you consider phototoxicity in your live fluorescence microscopy experiments? And good to see that we have an overwhelming yes on that one. And also a lot of people who are looking at it if it's obvious. So I think our awareness initiatives are paying off. I think it is something that people are becoming more and more aware of. So the question of only if it's obvious is our next question. How do you assess phototoxicity in your live fluorescence microscopy experiments? And I think we've seen this fairly often, that ensuring the sample morphology is normal is one that a lot of people look at. Measure and minimize photobleaching, and ensure sample dynamics are as expected, so like mitotic timing and so on. And then using dyes to assess sample health. And then we probably have a 30% or so who are not assessing phototoxicity or maybe unaware. And then if the live experiments are affected by phototoxicity, what do you do? So the majority will, I guess it's kind of a 50-50 split, with reducing the incident light intensity and increasing exposure time versus using a more sensitive or gentle microscope. So those are two great ways to minimize phototoxicity. So as I say, I'll make the report of this after the session. And so if anybody's still filling out the survey, please go ahead and we'll share the results.
Fantastic. Thank you very much. Okay. Right, let me get back to, okay. Excellent, good. Thank you for that. Okay, so yes, maybe to start with, after the questionnaire, one of the things that we're trying to do in the working group, and that always emerges as a bit of an issue, is that sometimes the terms are not all that clear. But let me just start with an example that is relatively famous within the community. But Hinchcliffe and Alexei Kodiakov and Greenfield's Kip Saluda and so on published a paper in Science where they said that the centrosome is required for cell cycle progression if you want the cell to go through G1 into S phase. Because they removed the centrosome by laser ablation, and they found that arrested cell cycle progression. And then 6 years later, with some of those same authors, they had a paper in JCB where they realized that cell cycle progression does not require the centrosome. There's just de novo centriole assembly of that, and they can progress through the cell cycle if you lower the intensity. And after that, it was then Magidson and Kodiakov who wrote a very nice review about circumventing photodamage in live cell microscopy. And if you have no idea where to start, then I really recommend that one. That's just a really really good read. That was for me the first one that really started to get me into this, and it's well written and entertaining. And I think it's also just that kind of lovely fessing up to, you know, having not been aware of that and then raising awareness through that. So one of the take-home messages was there to lower the amount of light that is used. The other thing is also that you can have this cumulative damage. And so removing the centriole through laser ablation already made for relatively stressed cells. And if you add then to that relative high intensity for fluorescence excitation, that just did those cells in. And so in terms of the definition of the terminology that we'll be using, I would here say that phototoxicity and in photo damage, they're often used synonymously, but what we're talking about really is the damage to cellular macromolecules upon excitation light illumination and its secondary effects, I might add there. And this can lead to the impairment of the sample's physiology. That can go all the way down to sample death. And that means that if we have live imaging, we always need to include there as well, not just that the 3 points of spatial resolution and temporal resolution and signal to noise ratio or signal to background ratio, but also sample health. And the way Claire and I see it is that often actually sample health needs to be put at the top of that importance pyramid, because you can otherwise end up producing misleading results. And that in turn means that you don't just have a yes or no answer or an on and off answer, but you have something that then is wrong. This is from train points in where they would say whether the train goes left or right, if you have that on or off, or if there's something wrong with that whole setup. And that is something that can be a little bit treacherous about phototoxicity, as seen in that first example. And so what we are trying to do in work group 13 is to look at all the different aspects that play into this. And that means that this can start with even just your sample preparation and the sample type that you are using. What is the label or fluorescent protein that you have? What is its localization? Is it in the mitochondrial membrane or in the cell membrane or just swimming around in the cytosol? What are the media that you are using? Certain media can lead to mitochondrial fragmentation and can simulate phototoxicity before you even put any light on your sample. One of the key things definitely is that it's important to be able to measure the incident light if you want to be quantifying this in an absolute manner. And playing into that is the type of microscope that you are using. And then there's a fair amount of technical considerations there as well, such as illumination overhead, which Claire will be talking about. And then another aspect of this is, how do you measure that effect? What is your biological readout? And as we saw, a lot of people will go for morphology. Dynamics are definitely also a good readout to use. And then what are the controls that we can use for this? And these might also highlight issues that are not just the incident light, but also, for example, temperature control and so on. How do we then quantify and report that? And that is an important aspect in terms of how should this really be measured and then put into publications. What is the image processing that we can use? Now, with the advent of AI, there is the possibility to reduce that even further, just using image restoration. And again, what are the controls, and can we have any kind of standards? And then really to share these findings. And it's not, it can never quite be perfect, but it's important to be able to point out what the potential limitations of a given study are, and to just be very honest and upfront about that. And so what we really want to do is raise awareness of this and then widen the participation of this discussion. So involve more research groups, image facility staff who are often the gatekeepers of good imaging, as that is passed down in terms of training to other groups. But we also are very keen to involve more instrument and software manufacturers in this, and then ultimately also to have scientific publishers and the reviewers of manuscripts and grants on board, and to be able to share many of these approaches that we recommend. For one of the more recent updates, I have a focal plane article here that we have hyperlinked, so we will make these slides available after this event, and all of these things are hyperlinked, so you should be able to directly go to those and have a look at that. In that article, we also do have a whole list of useful publications that we are using for this. Okay, and so just a couple of key pointers really before we start with this in a wider context. But one of the things is that people often think it is only if you have fluorophores in your samples. This is for us a reason to advocate the term phototoxicity and not fluorotoxicity, because you can have phototoxicity without any fluorophores. That's something that we demonstrated in Nature Methods commentary in 2017. And even just, you know, a very moderate amount of blue light, 4 to 5 minutes, can lead to this, to a reduction in proliferation. So even just moderate amounts for a relatively short time may already have an impact on your cells, and you don't need any fluorescent proteins within that. The other thing is that one of the key players, and possibly the most important key player when it comes to phototoxicity, are reactive oxygen species. We're using that as an umbrella term for singlet oxygen and hydrogen peroxide and all of these other species that are part of that. It doesn't matter necessarily so much which ones are relevant there, but it is often this as a secondary effect of the excitation light and then relaxation and so on that will lead to cells being affected by that excitation light and the secondary effects of that. Again, here we used a hydrogen peroxide sensor. in mammalian cells, and 5 minutes of very moderate blue light exposure immediately makes them light up like nothing on Earth. So this in turn means even if you're just looking for your cells using fluorescence, that may be a relatively bad idea, as you could already have an effect. One of the other things to point out very early on is that phototoxicity is highly sample dependent. Here a paper by Weltjen and co-authors, where you can see that, for example, HeLa cells are very robust, whereas U2OS or COS7 cells register much earlier an effect of phototoxicity. Equally, if you are looking at C. elegans as opposed to, for example, Drosophila melanogaster or so, that will have an effect in its own right. And then even if you are using the same cell line with the same kind of fluorescent label, you might have differing effects depending on the stage that they are in. Like dividing cells will have a much lower phototolerance than quiescent ones. And the corollary of this is that we cannot have a standard phototoxicity test. You need to adapt that to the cells in question and your particular sample in question. So very briefly, as I need to be aware of time, but we have there the definition of phototoxicity. Photobleaching is important as well. That is an umbrella term for light-induced driving of fluorophores into any dark state from which recovery is slow. It can also be the irreversible light-induced destruction of fluorophores. Important here as well is the measure of intensity, rather the physical term being irradiance, but very often in live imaging we use intensity. And that the measure of that is often be that milliwatt per centimeter square or variance thereof. So that really is just the power received by a given surface. We then have exposure time, and then we can have the combination of those two, which is then the dose of light. And a useful working model here is that one of the things that are good to avoid is really this accelerated, also called supralinear photobleaching, that happens at high intensities. We will be revising this working model as well, because it seems that in this, if we take the three components, duration of observation, the cell health, however we assess that, and then the intensity, as mentioned before, that if we are below a certain threshold, it is possible that it doesn't matter if we use higher or lower intensities. So there may be a safe range that we have there. There may be a positive effect that can have, but there is a certain range in which it doesn't seem to matter if it is lower or higher intensity. So there's a bit of a caveat to this working model at the moment. Claire will show you more about that. But this is based on some of the work in my lab, but also previous publications that I have hyperlinked there. And that will be very useful for you to consult if interested. Another thing is in terms of intensity, that very often is, well, it is a function also of the magnification or also the numerical aperture. Here just one numerical example that I produced with MATLAB, where if you have 10 times magnification, you have a relatively low intensity, 20 times magnification quadruples that, because you have that in 2 dimensions. And so the corollary here being the higher your magnification or your NA, the more likely you might be to induce damage in your sample. Okay, and with that, I would like to hand over to Claire to talk about phototoxicity sources.
Thank you. So we're going to sort of go through some different examples of where the phototoxicity can be coming from. So based on the survey, I think a lot of people try to minimize their excitation light. So a lot of the time we're used to trying to get really nice images of our cells, but if we have really beautiful images, but the cells are compromised, then they're not very valuable scientifically. So using shorter exposure times or lower incident light power is one way around that. And oftentimes, depending on the software we're using, we may not be using the dynamic range of our sensor. So if you look at the images shown there, we have the different exposure times for the same sample. And you can see when we get up to a 1 second exposure, we have a nice, bright, high contrast image. But if you look at the plot to the right, those are normalized intensity data. And you can see that even in the 100 millisecond exposure, we have the same intensity profile. We just have a bit more noise, but it's still something we can definitely measure and quantify. So going to that lower exposure time, even though the sample may not look as good on the screen, if you normalize it with that second panel of images, you can see that we still see all of the structures we're interested in quite well at the lower exposure time. Next slide. So one other thing is to minimize the number of fluorescent dyes or proteins. So we have so many amazing tools and resources out there. We want to look at everything, but just having those dyes present in our cells and then having that those imaging conditions for each of those colors can really cause a lot of issues. So if you look at the 2 videos on the left, you see the cells are migrating. We have a blue nuclear dye, red mitochondrial dye, and then a paxillin, EGFP, and focal adhesions. And you can see the cells are migrating nicely, but in the second panel in the middle of the slide, we actually look for a much longer period of time. And you can see that the cell division is inhibited and we're getting very strange looking nuclei. And really the cells are not happy at all. Whereas in the right panel there, it's the exact same sample on the same plate image at the same time. You can see that the cells are dividing and migrating and the nuclei look quite normal. And in the graph below, we looked at different imaging conditions, different buffers, and the cell tracking. And you can see just having the nuclear dye present actually slows down the cell migration quite significantly. Next slide. And something that we came across in around 2018 or so, we were looking at high intensity light for a short exposure time versus low intensity light for a long exposure time. And we had been convinced for a long time that it was much better to have lower light intensity over a longer exposure time, even if the total amount of light on the sample was consistent. And what we would find is if in the first panel, we use a 24 millisecond exposure, but with fairly intense light, you can see that the cell migration shown by the rose plot below is not as significant as if you go to the far right side where we have a 1 second exposure and a much lower incident light. The cells are migrating much faster and further. But what we determined during the duration of this study was that it was actually what we call overhead illumination, where the sample was actually being exposed to light for a much longer time than the camera exposure time. So if you want to go to the next slide. So during a live imaging experiment on a camera-based microscope, you basically open your shutter, you turn on your light source, then you expose your sample and take an image, then you close your shutter, save the image, and repeat that cycle depending on the time scale. And the key is that all shuttering is not the same. So if you have an older microscope with a bulb-based light source, you leave the light source on, and you usually use a mechanical shutter to turn on and off the light on the sample. And these shutters can be quite slow. And even with newer LED-based lamps, which can be turned off on and off electronically, we found that depending how we had set up our hardware and our software, if we used USB control of the light source from the microscope software, we had a much longer time of delay between the turn on, when the lamp was turned on, and when the light source would start collecting light. So we found if you set up a TTL trigger where you directly trigger the light source from the camera, then we could eliminate this delay or this overhead illumination from the shuttering. Next slide. So I'll say a little bit more about that after, just to show you the difference if we do eliminate that factor. The other one that Philippe talked about a little bit already is that longer wavelengths are better, and that blue light can be toxic. And so this is an example where they looked at cell colonies and they exposed them to light, and then they actually waited 7 days. So they let the cells grow for 7 days after the light exposure. And what they found was even without fluorophores present, the shorter wavelengths inhibited the cell division and the colony formation. So you can see in the blue data points there with 730 or 375 nanometer light that the plating efficiency or the colony formation was much reduced. Whereas with the longer wavelength 633 nanometer light, which has much lower energy, the colony growth was quite significantly higher. They also looked at Lorden, which is a lipophilic probe, which is excited at 391. And you can see now that we have the fluorescence process and more phototoxicity, that the plating efficiency decreases very quickly under those conditions. So there's different ways to look at the phototoxicity. So we're going to go through some of the different ways to assess it. So morphology, of course, is one of the ones that has come up already. Cell death is another one that you can look at. Organelle morphology, so rather than the cell itself, you can look at mitochondrial morphology or other organelles you may be studying. Developmental defects, if you're looking at an organism-based model. Cell division or proliferation, so how successful cell division is, and then what are the kinetics of that. And then there's some more sensitive techniques like looking at calcium signals or voltage changes in neurons. So the ability to measure the phototoxicity effect is really dependent on what you're trying to measure, and there's relative sensitivity to those different methods. So I always make the joke with cell death that even morphology, when the cell morphology starts to change, you're pretty far along the phototoxicity pathway. And I always make the joke, if you were doing a clinical trial and all the patients were alive when they left after their treatments, and you say that the drug is good, nobody would be too happy about that study. So just looking at cell death or morphology is better than nothing, but it's good to start to look towards more sensitive methods. Next slide. So photobleaching is one way that you can look at this to get a sense of how the light is affecting your sample. And this is an example from our paper where we looked at this illumination overhead. And on the left side, it's that same experiment that I showed you with the cell migration, where we looked at different intensities of light and we adjusted the exposure time. So the sample was getting the same total light dose. But in fact, it wasn't getting the same total light dose because we had this delay with the shuttering. And so you can see as we go to higher intensity, the photobleaching was much higher. So these are fixed samples just in a PBS buffer, so that we don't have any anti-fade agents or anything. So mimicking our live sample conditions well. But then on the right side, when we eliminate the illumination overhead by directly triggering the light source, you can see that difference in photobleaching disappears. And you can see the scaling is different on the right curve. So the photobleaching overall is much reduced. Next slide. Another thing we can look at is reactive oxygen species. So Philippe showed one example of this as well. This is a probe called Cell Rocks Deep Red. And in the first image you see there, the dye is actually dark when there's no reactive oxygen species around. And then we did 400 images of a GFP protein for 100 milliseconds at 21.3 milliwatts. And you can see that we see the ROS generation there. And you can see on the graph on the right that there's a power dependence. So the more power we put in, the more ROS we generate. So this is a good assay, which is going to be much more sensitive than just looking at cell morphology or cell division. But we also put in the caveat that you really should do this at the end of your experiment, because if you're imaging this ROS dye throughout your experiment, that dye and the image of it can also add to your phototoxicity. So it's good to do it at the end. Next slide. And you can also look at mitochondrial morphology. So this is going to be much more sensitive than cell morphology. And you can see in the top panel, we have this nice mitochondrial, these spaghetti-like structures within the cell. And in the bottom panel, when we have high light doses and illumination overhead, you can see that the mitochondria condense. And in the right side on the graph, we look at quantifying that just by looking at mitochondrial area. Next slide. And this is a paper I always love to point out because, as I mentioned with the clinical trial, you want to look at what happens to the patients after the treatments. So this is an example where in panel A, they look at an area of yeast cells that were not imaged during the experiment. And they look at the cells for 24 hours after the imaging. And you can see that the yeast are dividing and multiplying quite rapidly. Whereas in panel B, that those cells were imaged with fluorescence imaging for a period of time under conditions that didn't appear to be toxic or problematic, but you can see that in that panel, which is right next to panel A, the cells are not dividing. So there's been some phototoxicity that wasn't apparent during the imaging experiment. Next slide. So I'll turn it back to Philippe.
Okay. Just a few words on photo bleaching. That, of course, that will depend on the microscopy method that you use, and that is from Harry Schroff's group, a nice example where they compared the dye spin to a spinning disk. And even though the spinning disk is relatively mild, it's not as shall we say brutal as a spot scanning confocal, it still doesn't, it pales in comparison literally to the dye spin. And so photo bleaching is a way to assess that. It is an indirect way, and as Claire said, just because you don't have photo bleaching doesn't mean you don't have phototoxicity, but it is one way to check for it. And if you have rapid photo bleaching, usually that means your cells are not, they're going to somewhat suffer from that. An example from Magidson and Kodyakov's review, where you see rapid photo bleaching in example A, and a lot of blebbing in example, in the bright field, DIC image underneath that. And lowering the intensity leads to reduced photo bleaching. However, you still are getting blebbing there. And so what they had to do was then increase the dark intervals between illuminations to 5 minutes, and they ended up then having no blebbing. But that is one reason why taking an optical contrast image or a bright field image is very useful, because that comes at no additional cost for the cell, but you can have a look at the morphology. So that tends to be always a very useful control. And while photo bleaching is not ideal as a measure, sometimes it's the only thing that you can do. This is from my group, a coral paper where we were imaging live corals and tried to have the fairest or even the kindest comparison of light sheet versus wide field microscopy. And it turns out that even if you give wide field all the chance you can for that to reduce exposure, it still cannot really measure up to light sheet because it's just inherently, light sheet microscopy is inherently a very gentle method of doing that. But in terms of the behavior, there was nothing that we were able to compare in terms of dynamics. They didn't show differences in dynamics. So the only thing that we had was to go by the rate of photo bleaching. Another thing to do to use for assessment is a dose response curve. And here we looked at blue light intensities and the effect they have on motility here of a prostate cancer cell line. And you establish a dose response curve, and interestingly, we found a positive effect that we have with these as well. So it may be that you not necessarily negatively affect your cells, but that you have a positive effect, but that is still something where you are disturbing your observation. So that's useful to take into account. But dose response curves are very useful to get an idea of what the effect is of your observation. Motility, this kind of dynamic thing, is good to assess. When we did the same thing comparing mitotic delays, we didn't get any kind of significance. The black bar here being 95% compatibility interval. And so that was, mitotic delay was not significant, even at relatively high intensities, whereas motility picked that up. So does that mean that motility really is one of the most sensitive assays? Well, you can always go more sensitive. So one of the things we did in that same paper was to use just very moderate blue light illumination for a short time. And we didn't see any difference in the motility there. But then by using qPCR, picked up on big changes in terms of gene expression. Of course, gene expression is not the same as translation, but it is just an indicator of the effect that you might be having. And so you can always go more sensitive with the assessment. But one of the things, and we absolutely recognize that, is that not everyone wants to go to that length of assessing a potential effect that you're having with your observation. And so there is a bit of a trade-off that you can have in terms of how sensitive is your assay versus how simple is it to perform. And so live dead assays are very popular because they're easy to do. And morphology is an easy thing to get a handle on. Whereas motility can be a little bit more effort to measure. If you have cell protrusion rates or mitochondrial fragmentation, that can involve a fair amount of image processing that you need to do to be able to assess that. Using biosensors, you need to create a transgenic line. Not everyone wants to do that. So there is a bit of a trade-off with that. Having said that, there are some assays that are simple but also sensitive. So using bright field or optical contrast to look at mitotic delay, or then your long-term controls, what Claire showed with the Carlton paper, doing that after the exposure and looking at the longer-term division / proliferation of your cells can be a very good way of doing that. That doesn't really take that much more effort for experimenters to do. Okay, and so really we're getting to the point of what are our possibilities to reduce phototoxicity. And as we pointed out now already a number of times, having the right type of microscope is important. I mentioned spinning disk before. Actually, wide field can be a much gentler way of doing that, because we have the micro lenses in spinning disk confocals that focus the light, and with that we have higher intensity. Generally, reducing the intensity and having a lower light power does seem to be a good way to go forward. If in doubt, you can do a dose response curve with that. This is the very nice paper by Tinves and Spencer Short and others, where they use C. elegans development to assess that. And so choosing the right microscopy method is very important. And so it's very encouraging to see that many of you, if you do find phototoxic effects, that you will try and switch to a gentler way of doing that. Here, just a very brief cartoon to show you why light sheet is gentler than many, or pretty much all of the other methods, because we are only illuminating the focal plane that we are imaging at that time. And so with conventional, where you have illumination and detection through the same objective, for each focal plane, you are illuminating the entire sample. Whereas in light sheet, you are only doing that for each focal plane. Now, if, as in this cartoon, you just have 3 focal planes to worry about, that doesn't weigh in that heavily. You have a 3-fold reduction with the light sheet. If, of course, you are doing something larger like Drosophila or at very high resolution, you can have, 100, 200-fold reduction, and that will have a big effect of that. So the microscope you choose has a very important effect on that. And so all of these things taken together will then really have that big impact on your observation. What is the sample? Use a minimum number of dyes. Use long wavelength dyes if possible. If you can have stable transfections instead of transient ones, we have a better control of the number of copies that you have in there, will be useful. Generally, longer excitation wavelengths do seem to work better, bearing in mind the caveat of Claire's research and also the Andreas Cuny paper. I will show that reference at the end. Pulsed illumination may be very effective. The jury is still a little bit out on that. I think we need more quantitative publications on that to really be able to absolutely be sure about that one. Certainly light sheet, just going for the focal plane, and then eliminating illumination overhead is a big important one. Detection wise, always go for the most sensitive detection that you can have. Reduce your background, so that will in itself give you a better signal to background ratio. High transmission optical components, the fewest possible optical components, and you can use camera binning as well. And then having the right controls is always very important also in terms of being able to show that you have minimized that as best possible. Okay, we have mentioned this before here, also hyperlinked to a very nice review by Icha and co-workers. And if you can, always worth shifting towards the longer wavelength. Here I will give over to Claire because that really is her area of expertise.
So I was mentioning how this overhead illumination, or this extra time that the light is on when the camera is not collecting fluorescence, you can see in the first panel there, when we use high light intensity for just a 24 millisecond exposure, in this setup, we had 113 millisecond illumination overhead. So we were really exciting the sample 4 times longer than we realized. Whereas in the second panel, we have the same amount of light, but we've eliminated that illumination overhead. And you can see that the cells are migrating in a similar way as they do with the lower light levels, or even with bright field. So one thing we've found from this study is that every microscope is different, and it's really sensitive to how you set up your hardware and how you set up your software. Even just going from a CMOS camera to a CCD camera has a big impact, and the different manufacturers handle this differently. So we're actually working on a protocol, which we're going to publish pretty soon on Protocols.io, just showing people how to measure it, because we think every instrument is different, and you really should just measure it on your own and make sure you find a way to eliminate it. And as long as you have a triggerable light source, like most solid state lasers and LEDs, you should be able to set it up to eliminate that. But a lot of times people aren't aware, including the manufacturers. So this is one thing that that can have a big impact if you're aware of it. Next slide. I know we're nearing the end of our time, so I'll maybe go through these a little bit more quickly, but we're also happy to share the slides. So if you just want to advance, Philippe, so the second panel comes up. This is just showing how, depending what you're measuring, you can bin the camera. So in the top panel, if you're just trying to track cells or measure fluorescence intensity of your cells, even using 4x4 binning is fine. And you can really reduce your exposure, the light that you put on the sample by adding together the pixels. But then in the bottom panel, if you were actually wanting to look at the adhesion structures within the cell, then the 4x4 binning is a little too much. So you'd probably have to go with 2x2 or lower. But it's another good tool to use. The next slide. And then this slide just shows when you're actually just finding your sample on the microscope, really make sure, if you can do it with bright field, that's better. You can dark adjust your eyes if the room is bright or the monitor's in your peripheral vision. This shows just 50 seconds of, maybe if you can go back up and down again, Philippe, these aren't looping, just 50 seconds of exposure time on the microscope, and you lose about 80% of your light if you don't attenuate the lamp. So yeah, there you go. So those are at the same time scale, and you can see with just putting a neutral density filter in to reduce the exposure light, just while you're looking around the sample, really removes a lot of that problem. Next slide. So, so we have a bit of a summary here. I'm not sure, I guess we can go through it in fairly high level since we're 3 minutes left, but one of the things I think we wanted to set expectations for the community, that anything is better than nothing. So if people just start talking about it and thinking about it and monitoring whether this is a problem, measuring it if you can, and then it's really important to report it. So in your paper, you should say what you did. And if you didn't do anything, it's okay. It may depend on your model system or what constraints you have, but you should report that phototoxicity was not taken into account, and then just present your results in the context of that. Next slide. And then, as I mentioned, with the illumination overhead, every system is unique, as Philippe was talking about, with how the light is delivered has a real impact. So things like light sheet are really great for minimizing phototoxicity, and then something like STED is going to have a lot of of things to take into account because the light is focused and you have a depletion laser. And then every biological system is different and has different sensitivity to the light as well. So I'll turn it back to Philippe to wrap up.
Okay, so yeah, just briefly, if it does pay off to start with a good dose response curve, and then you may have to make a bit of a compromise, but there will be. It is important that you can be clear on what are the parameters that you need to observe, what is the process that you are keen to have, and vary the power a bit. And one of the things that Claire and I and Working Group 13 always say is we should step back a bit from trying to have the really pretty high contrast images. If you have a signal to background ratio of 4, that for most image processing steps that we have nowadays is good enough. And it's not about the pretty picture. It really should be about the happiest possible cells. And so there is this trade-off between signal to noise, a signal to background, and then the power that you are using and the cell health. And the other thing is, do bookend your cells and damage them and get an idea of how they look when you are damaging them. That will give you a good idea of what to look out for. So do knock them on the head, and then try and reduce that as maximally possible. And so really, there is a bit of a recipe that you can use for that, where you say, what is the temporal and spatial resolution of the biological process that you are interested in? And then you can say, okay, what is the minimal contrast that you need to be able to process this, to do image processing on a reliable and automated or semi-automated scale? And do this image processing before you acquire tons of images. Do a test round, and then establish with that a dose response curve and find what the most sensitive biological readout is that you can use with the data that you are acquiring. And morphology is okay, but usually dynamics tend to be a more sensitive kind of figuring that out. And so you reduce that as much as possible while still getting images of sufficient contrast. And so that really is a principle that is used in radiation biology, which is as low as reasonably achievable. And by that, we're also acknowledging that you can't completely eliminate it, but at least reducing it is always definitely worth it. And then to have controls in there, which we've mentioned now a number of times, is very, very useful. Also, to convince reviewers of the paper that you have submitted. Okay, this is another summary slide, but I think really we are now at the end. Here are recommended reviews that we have. That focal plane article, which has more of those, and then some of the primary articles that I mentioned, also the Schmidt-Kuhn and Rudolph paper here at the bottom. So please have a look at those and check those out. And we will make our slides available. Yeah, here are just some light sheet microscopes that my kids made a few years ago, but the illumination from the side. If in doubt, use light sheets if you can. Right, so we will have maybe a minute or two for questions. Sorry, we were a bit ambitious in terms of the amount of slides and the time we spent on those. But does anyone have questions at this point that we might be able to answer? Thank you very much for attending, and thanks for your attention.